So as they mentioned, I'm actually from the University of Wisconsin. Some of you may have some connection to Wisconsin. If you don't, Wisconsin produced Tony Romo, quarterback of the Dallas Cowboys, which may be a good thing, may be a bad thing, depending on your point of view. So anyway, but it wasn't my fault, so I didn't deliver him. But anyway, my job here is to actually give you some updates on some of the other disease states. Dr. Scheer gave us a really nice introduction to that. As he mentioned, I'm not a neurologist, I'm a basic scientist, and about several years ago when I started my lab at Wisconsin, I started studying how peripheral nerves are formed and how myelin is made around them, and we're very much interested in the genetic regulation of that. And about 5 or 6 years ago, I was really interested in thinking, well, some of our discoveries actually have some impact on CMT, and so I was in the beginning of the process of trying to develop some grant applications, which would take a year or two to do that. And lo and behold, I got a phone call from the CMTIA, actually Mike Schei, and said, we'd like you to develop some assays to identify compounds that would treat CMT1A. And I said, oh, really? Because I'd like to do that. I was really preparing to do that. And he said, well, we want you to start next month. And after I fell down and picked up the phone again and got back up, I was fortunate to get involved in this effort, and I've been inspired by Pat and Susan and all the members of the organization. I'm also inspired by all the patients that come to gatherings like this as well. So since then, I have also become a board member of the CMTA and part of really a large effort, which I'll tell you about. I've gotten so involved, I gave Pat my cell phone number, which I'll always regret, but he's kind of cut down a little bit, but we have a lot of great things to talk about. So part of what I want to do is communicate my enthusiasm, because it took a lot of efforts to get where we are, but I'm really excited about that. So basically my call here is to give you some updates on 3 different forms of CMT. CMT1A, which affects the Schwann cells that Dr. Scheer mentioned about. This is referred to as a demyelinating form. And then 2 other forms, which are on total 3 of the most common 4 types of CMT, which includes CMT1B, which is also a condition affecting Schwann cells, and also CMT2A, which is an axonal form. So the problem is in the transport of these nerve impulses. So this is an axonal form of CMT2A. So I want to, this is a tall order, but I'll try to give you pretty good updates of where we are today. So CMT1A itself has been the focus of my efforts to date. This was actually one of the very first causes of CMT that were identified a little over 20 years ago. And this resulted from several studies by many groups, including Uli Suter, that identified that this was not actually a point mutation within a single gene that causes it, but rather you have a duplication. So there's a segment of this chromosome which essentially gets duplicated. Sometimes you read text and see a duplication of a paragraph. In this case, we're duplicating a gene, and this is a very important gene known as peripheral myelin protein 22, PMP22. And as it turns out, this duplication turns out to be the most common cause of CMT. It accounts for at least 35% or a third of cases of CMT, at least where we know the genetic diagnosis. And in some surveys, it's maybe up to half or so. But we know that PMP22 is really an important protein. It's a part of this chocolate cookie part of the Oreo that Dr. Scheer was talking about. And it's embedded in this myelin membrane that wraps around these axons. So essentially, what we have then in CMT1A is that normally we have 2 copies of a gene, one from mom, one from dad. In this case, there are 2 copies of PMP22 gene on a single chromosome, giving rise to 3 copies in total instead of having just 2. And we think we know that that is the cause of CMT1A, and one of the reasons that we know that is that a lot of work by a lot of groups have shown that if you introduce an extra level of PMP22 into rats or mice, those rats or mice really begin to have some behaviors and symptoms that are sort of mimic what CMT1A patients experience. So this is a problem. It's not rocket science. I wish I was more complicated. I could try to really impress you, but it's actually relatively simple. So you have 2 copies of the PMP22 gene. Each gene makes a protein. That level of that protein, we'll call that 100%. In CMT1A, we actually have 3 copies of the gene. So we just have too much of the protein that comes from that gene. And so our job really is just to kind of turn down the volume, so to speak. Turn down the level of PMP22 to bring it back down into normal levels here, right at 100. So we don't have to turn it down very much. It's a little too loud. We need to back it off a little bit. And we know from studies that many people have done throughout the CMT scientific community that reducing the levels of PMP22 can be used to, at least in rats and mice, to ameliorate the symptoms that they experience, and therefore has great promise for humans with CMT1A. So the question is, how do we do it? And we've been pursuing essentially approaches to try to identify small molecules or compounds or some kind of drug that would be able to do that safely and effectively for CMT patients. So I want to tell you kind of a little bit about the process by which we do this. And it actually starts with sort of something we see in Wisconsin this summer, the humble firefly, which we see in the summer. I don't know, do they have those in Texas? We always have a debate whether we call them lightning bugs or fireflies. I don't know if Texas is a lightning bug state or a firefly state, but essentially a company in Madison, Wisconsin has developed products to take advantage of the particular enzyme within these flies that actually produce light, that beautiful glow that you see in a summer evening. And this enzyme is referred to as luciferase. And I know it has kind of a dark name. Lucifer, refers to the Lord of Light. That's the origin of the name there. And it just means that this is a gene that produces light. And essentially, this light can be used as sort of a way of being a meter on certain genes to see how active those are. And basically, what we've been trying to do is harness this particular enzyme, the gene that produces that enzyme, and hook it up to the PMP22 gene so that we can read out how much PMP22 is being produced. You know you have like a gas meter on your home. You can tell how much gas or electricity you're using in your home. It sort of reports the level that you're using. In this case, we're using a light assay to tell us how much PMP22 gene is being produced. So our job was to try to create then a cell-based assay where we have this gene hooked up to PMP22, and we call this a reporter. So it's reporting or metering, telling us how much PMP22 gene is being produced. And this becomes a very easy way then to determine what the level of PMP22 is. If we can hook that up appropriately, the amount of light we see kind of tells us how high PMP22 is within a given cell. So we took advantage of some really new technology that's come about in the last few years. And I won't have time to go through this, but there's a lot of excitement about a technique known as genome editing. And genome editing is a technique that can be used to very easily kind of modify the genome. In our case, we were trying to take the PMP22 gene and then hook up this luciferase gene right to it to create that meter that we were talking about. And therefore, this technique is really exciting. There's, in fact, some efforts around the world to maybe think about doing some sort of gene therapy with this. But essentially, it allows you to think of the genome like you would a word processor document. You can go in and change periods and move text around. This is the same thing that you can do now in the genome in cells to create that. So we were the first ones to actually do this, to put this reporter assay into a cell like this and to actually perform a drug screen. And we did this last couple of years with the help of some collaborators at NIH, which I'll tell you more about in a minute. But essentially, this is how this works. We're taking Schwann cells. So these are the cells that make myelin. And they've been modified in the way I described to have the luciferase gene right within the PMP22 gene. So it's reporting the level of PMP22. And you see this plate here with like 100 or so different wells where you can drop cells into those wells. And basically, each of those wells, since they have the luciferase gene, they can emit light in proportion to the level of the PMP22 gene. And then what we're doing is adding different chemical compounds to each of those wells of the Schwann cells. And then we're looking at which of those wells have a certain compound that lower the level of PMP22, going from 150% down to 100% ideally, or somewhat more. And then basically you can see these 3 wells here that have a little bit less light than the other ones. These particular compounds would be then candidates for those that can reduce the level of PMP22 and bring it back into line. And then ultimately these candidate drugs are among those that we pick and then pick for further testing in other assays, other tests, to see if they're safe to cells, that they don't kill cells, and that they would maybe have some promise for CMT1A. So I'm showing you at a level about 100 different compounds how this process works. And this was actually developed in collaboration with a lab at the NIH in Bethesda, Maryland. This is a lab that's directed by Jim Ingles, who's been sort of a leader in technology development for drug screening. And he works within a part of the National Institutes of Health, which is called NCATS, National Center for Advancing Translational Sciences. And basically, this is really a state-of-the-art drug screening facility, which has been dedicated to finding new drugs for disorders that currently have no treatment, like CMT. And so essentially, we initiated this process to take those assays that we developed and do the process that I just outlined, and actually then do a particular drug screen. And what this film is showing here is these little robots here, which similar robots are used in automobile manufacturing to do these sort of precise assembly movements. Well, in this case, these robots are programmed to move plates around. And these particular plates, you can see them in this little carousel that's rotating. The robot is taking these plates out and putting them in different machines. Each of those particular little plates that you see has 1,536 wells. Each of those have Schwann cells that are making light. And to each of those little wells, then a little bit of a certain compound's been added to see if they can reduce the output of the PMP22 gene. And so one of the reasons I'm incredibly excited is that just this last winter, they were actually able to screen up to 400,000 compounds. They did each of those compounds at 5 different concentrations. So that's essentially 2 million different assays they were able to do. And thankfully, we're not asking people to do this because it'd be incredibly boring, but these robots don't get bored very easily, so that's very helpful. And essentially, we're done with that. And then as Pat has mentioned, we're also extremely excited about the fact that we formed this partnership with Sanofi Genzyme, and they just recently finished 1.9 million compounds. And these aren't the same compounds, so Sanofi Genzyme has their own proprietary kinds of compounds that they've developed over years. These include all sorts of things. They include like natural products that are isolated from rainforests everywhere around the world. There's a whole, you can't even imagine the number of different kinds of sources that they have as well. But between these two sources, we've actually screened close to 2.5 million compounds, which is just mind-blowing to me. Now, I want to tell you that certainly this is a major accomplishment for us. And right now, we've identified somewhere to 10 to 20,000 compounds that have passed out of these screens here. This is a competitive process. This is a free market. We're asking these compounds to compete against each other to be the most effective and safe ways of reducing PMP22 concentrations. But we have a lot of work to do to take those 10 to 20,000 compounds and then funnel them further down to identify those that are really going to have the most promise for treatment of CMT1A. So how do we do that? How do we determine if drugs are affected for CMT1A? So what I've been telling you about is about cell-based assays, and there's several others that we've developed. I want to tell you about a couple other approaches that we've invested in. One is testing compounds on myelin that is made actually within a dish in a laboratory, and also tell you a little bit about how we are taking advantage of rodent models of CMT1A as well. And in fact, the CMT has licensed really two of the best rodent models of CMT1A, which are the C3 mouse and CMT1A rat. But let me first tell you a little bit about looking at myelin in a dish. So this maybe doesn't show up too well, but one of our network partners, our star investigators, is Laura Feltry at the University of Buffalo. And she's using a system in which we can take neurons, which are normally what Schwann cells make myelin around. You add Schwann cells, and under the right conditions, they actually do form myelin in a dish. And you can imagine that if you can do this in a culture, it's a lot easier than having mice or rats in terms of at least the numbers of tests that you can do. And these little threads here that are green indicate axons that have formed from neurons. And this green signal here indicates that those axons are covered with myelin. So we can use this assay, we can use the drugs that we identify in these screens that we've done to test and make sure that they're safe, that they don't make myelin get worse, and that they can improve myelination as well. So this is an important sort of downstream validation of our initial screening assays. I did also mention the rodent models of CMT1A that we have, and in fact, we've housed these within a really state-of-the-art testing company known as Psychogenics, and they can do various tests on mice, and so when we use compounds and test them in mice, we can not only look at PMP22 levels, but we can actually test if the motor function is actually better. And what I'm showing here is actually one of the tests that they do. This is sort of a balance beam, sort of the Olympic test for mice. But this test is essentially a mouse has to run up this balance beam and do it quickly and not slip too much. So you can see this mouse is kind of running up there. It's actually pretty good. It slips there once, but then it reaches the top. So in the mouse model of CMT1A development, they look a little different. So this is that particular mouse that has a good sort of resemblance, I guess, to CMT1A. It's running up the balance beam. It's a little slower. You can see its hind limbs are kind of slipping off a little bit. It's having trouble staying on the balance beam. It finally does reach its goal. And so when we develop these compounds, we can simply ask is if we treat mice like this, are we able to make them better at actually traversing this balance beam? So this is just one of the tests that we can do. But it's a very important test to be able to assess that our compounds will eventually be effective in this model and then hopefully also for patients with CMT. So aside from those tests, Pat already described that we've invested heavily in stem cell models for testing. And very importantly, we want to be able to test compounds on human Schwann cells that come from these human stem cells that we've done. And Pat already showed this process here. Basically, we've isolated stem cells from the skin of patients with different types of CMT, and then we differentiate them into Schwann cells in a dish. So now that we have human Schwann cells that we can use for testing. And these are just some pictures from one of my colleagues at University of Wisconsin, Anita Bhattacharya, who has made Schwann cells in a dish that have the right markers. That's what the colors indicate, green and red. There are just colors indicating that they are actually successfully Schwann cells. And I also showed a picture from Gabsang Lee, who has also made Schwann cells. And in fact, he's one of the leaders in the country in terms of developing Schwann cells from stem cells. And in fact, is also doing the same thing from CMT1A patient cells, so they can look at those in a dish, differentiate them as Schwann cells, and then use those for the testing of different compounds that we have. So this is an important component of really evaluating the different drugs that we have. So I want to summarize everything that I've been telling you about in terms of all the pieces that we feel that are important for really reaching our goal of a clinical trial for CMT1A. So we've assembled drug screening assays, rodent models, stem cell models, and clinical outcome measures. So the drug screening assays, at least the first round of assays are done, over 2.3 million compounds screened. We have two of the best rodent models of CMT1A housed within cytogenetics that are being tested with compounds now and are available for future tests. There have been stem cell lines created for CMT1A, and we're making several more in collaboration with the New York Stem Cell Foundation to bring those online for further testing. And I said CMTIA is also investing in making sure that those Schwann cells that we make from those human cells are really good quality. And one thing that I've learned about this process is we have to really think backwards from what a clinical trial is going to look like. Ultimately, none of these pieces matter unless we know that we have ways to evaluate whether compounds are effective in CMT patients. So Dr. Scheer and many of his colleagues have really worked very hard on developing neuropathy scores and other tests that indicate the severity of a neuropathy and ultimately we use to assess effectiveness in clinical trials. So ultimately we rely on the patient community, you to be involved, and we also have an incredible international network of neurologists that are dedicated to CMT, which will be able to supervise future clinical trials. So I'm going to shift gears, talk about CMT1B. This is also one of the top 4 common causes of CMT. This is actually a mutation in a different gene known as myelin protein 0. And one of the things that's unusual about this disease is that basically the mutations in that gene can actually have very different consequences depending on the mutation. And I borrowed some descriptions from Dr. Shai on our board to indicate a couple of different descriptions. So one patient with CMT1B had a very early onset, actually only walked after the age of 3, required a walker or wheelchair. So this is a sort of an early onset case. Whereas a mutation in the same gene occurred in this person who only began to experience symptoms in their 40s, actually only then experienced some mid-weakness and then ultimately was diagnosed with CMT1B as a 50-year-old. So one of the mysteries there is to sort of understand why these different mutations cause these very different kinds of outcomes. So myelin protein 0 is a lot like PMB22 in that it's important for the structure of myelin. As Dr. Scheer mentioned, this is the Oreo cookie here of the axon here with these wraps of myelin around it, being the chocolate. And these wraps are very nicely organized. This is really great myelin. But in the absence of myelin protein 0, you get this sort of unraveling of all these wraps. And it begins to look a little bit unentangled and more tangled, and certainly not as effective myelin as this on the left. So we know MPZ is very important for this. And so multiple mutations in myelin protein 0 cause peripheral neuropathy. Why is that the case? The field has really made a lot of progress in understanding that the mutation in myelin protein 0, or many mutations, causes this sort of cellular stress pathway. It's also known as the unfolded protein response. The cell actually has quality control measures to make sure that proteins that are produced are folded properly and are able to work in the cell. And this is an example of another kind of light-based assay where you have a cell that has myelin protein 0. The myelin protein 0 is labeled in red here. This is another cell that is expressing a mutant form of myelin protein 0. And now you see this big green thing here. And that big green thing is just a signal that the cell is experiencing this unfolded protein response. And essentially, we now have proof of principle data that at least in some cases, if we kind of reduce the stress level, reduce this sort of green signal here, that would actually allow the cell to cope a lot better and hopefully to improve myelination. This is another area of CMT where we're extremely well equipped as an organization to deal with. Dr. Shai and Dr. Rabbits, who are members of our scientific advisory board, are two of the leaders in this area who have co-developed 2 mouse models of CMT1A-B. This is a mutation in one particular amino acid and another amino acid as well. And both of these mice are, in their own ways, excellent models of CMT1A-B. And they've identified this role, the cellular stress, as a factor in causing myelination problems. And in fact, they've also identified types of compounds that are able to alleviate, at least partially, the source of those myelination problems. In fact, Dr. Shai has some proof of principle data with a derivative of curcumin, suggesting that it has some promise for therapy, and then he's in the process of planning out and thinking about clinical trial with that. But that remains to be seen, but when that will occur. But nonetheless, these are areas where we have extremely good mouse models of CMT1A-B. So similar to 1A, I just want to tell you where we are with 1B and these different pieces that we need to put together. We have drug screening assays. We were assays that were developed by these board members, Dr. Shai and Dr. Rabbits. And then we have other versions in progress. There's someone at NIH who's supported by CMTIA in our partner laboratory who's working on creating better versions of these assays, because we appreciate that even though we have identified a couple candidates, that we really need to identify more candidates that probably have more likelihood of ultimately being a good treatment for CMT1B. We have 2 excellent models of CMT1B, and a 3rd model is in progress, which would represent the late type of onset of CMT1B. So I think between the 3 of these, we can sort of represent the spectrum that CMT1B patients experience. And in fact, stem cell lines are being created for CMT1B. As Pat mentioned, the lines have been sent to the New York Stem Cell Foundation, and we expect them to complete these cell lines by sometime this summer. And essentially, this is an area where we have synergy within the organization in the sense that we need to be able to develop human Schwann cells for both CMT1A and CMT1B. And because we were working on these 2 projects simultaneously, they can cross-fertilize and create this synergy of different approaches that we can learn from each other as well. So all the stem cell investments that we're doing are really not just targeted for CMT1A, but also the other subtypes of CMT as well. So finally, I want to turn to CMT2A. This is actually a different form of CMT that affects primarily the axons of motor neurons that project out to muscles, or also the axons of neurons that come back and communicate sensation back to the central nervous system. And CMT2A is caused by a mutation in this gene known as Mitofusin 2. And so you may have heard about mitochondria. These are the important energy-producing organelles within the cell. I used to be able to impress people with my knowledge about them, but last night a 5th grader at our table was communicating everything about mitochondria that's important to know. And my own kids are learning about this in junior high, so I can't impress people anymore. But she was a very bright 5th grader, I have to say, though. So, mitochondria comes from the word thread, mitos, and the word grain. Essentially, mitochondria are these organelles within every cell that are very important for producing energy. And when I learned about them, they were always these little static things that were just dots on a page. But one of the things that people have understood is that mitochondria actually have kind of a life of their own. They move around, they can fuse, they can divide, and the mitofusins are involved in this process where 2 smaller mitochondria can actually join together to create 1 larger mitochondria. And mitochondria are constantly doing this to serve the needs of the cell, and they can fuse and they can actually divide later on depending on the circumstances. And these dynamics of mitochondria, whether they're moving around or fusing or dividing, are very important for their function. And so basically there's some slides I borrowed from Dr. Balo at Cedars-Sinai talking about the fact that if you think about an axon, so the cell body is back in the spinal cord, and it's projecting its axon all the way to its target tissues here, the muscle, for example. These mitochondria are these little football-shaped things, and it turns out that they're very important for the axon. In fact, the axon is really highly dependent on having energy to maintain the strength of nerve impulses as they go out to a muscle, as well as to maintain their structure over time. I know that many of you, if you drive to El Paso, you know you're going to need some energy along the way, whether it's gasoline or caffeine. You'll need to get stopped somewhere along the way to make it all the way to El Paso. And so you're going to need to have some place to get that energy from. Well, mitochondria are great because they not only can serve those energy needs, but they actually move around and serve the function of those axons. So these axons are actually transporting not only mitochondria up and down, but they're also transporting other things either towards the target tissue or away from the target tissue back here. And this is the normal case of a healthy axon that's being able to transport things. But in fact, this transport is really important because if the cell experiences anything kind of toxic, in this case, make a little spike of calcium here, these mitochondria are helpful in protecting the axon in terms of its ability to survive and the result. But CMT2A axons are a lot less dynamic. The mitochondria are a lot less mobile. They're more likely to sit around and not move around too much. And that makes them vulnerable. So if you have an influx of something like calcium that comes in, this is much likely to cause more of a big problem. And in fact, they can cause the end of the axon here to sort of die off, essentially. And therefore, you lose this connection between the neuron back in the spinal cord to its target tissue, if this is a muscle. And so that's what makes this an axonal neuropathy, is you lose these connections to those target tissues. And so our job is to try to figure out a way if we can fix this. And one of the ways that we've come up with, based on work from Dr. Balo at Cedars-Sinai, as well as others in the field, is the fact that Mitofusin 2, just like any sequel, that means there was actually an original movie to begin with. So Mitofusin 1 is also a similar protein that does exactly the same thing. And he was able to show that if you express Mitofusin 1, it can compensate for this problem when you lack Mitofusin 2. So normally these mitochondria are moving around, but when Mitofusin 2 is lost, they stop moving around. But if Mitofusin 1 comes in, that re-stimulates that process, and you have healthier mitochondria again. And so that's one of the strategies that we're pursuing at the moment. One of the other things I just really wanted to highlight as a recent success of ours as well is the fact that one of the challenges that we had for CMT2A is that even though we knew what the cause was and knew some of the molecular problems and cellular problems, one of the things is we didn't really have a very good rodent model for CMT2A. And in fact, there have been multiple attempts, some within our network and some from other people, where they tried to engineer mice to contain mutations that are derived from CMT2A patients, mutations in MFN2. But they were all relatively disappointing, had very subtle defects. So we decided to attack this and really take advantage of new technology. So together we decided to utilize a company in St. Louis, again using this sort of genome engineering approach to engineer a mutation into a rat. And we're able to create a new rat model for CMT2A based on one MFN2 mutation, which is already done, and another one which is also done, although we're just beginning to get enough rats to look at right now. But what's incredibly exciting to us is that despite all the failures that we had in mice to create a good model, this rat has turned out to be a pretty darn good model of CMT2A. And this is the first time I get to show Steve Shearer's data while Steve is listening to it. But Steve is an expert in this area of neuropathy, and he's been assessing the quality of axons in these models. And you can see by now, based on your education in myelinology, is that these axons here look pretty healthy. They have really nice myelin rings. You have very kind of tightly linked axons. Whereas in this rat at 8 months of age that is derived from a CMT2A associated mutation, you've lost many of these axons. You can see there's very few axons. The few that are left look kind of sick, and they're probably on their way to dying. And so at the cellular level, this is actually a pretty good model of CMT2A. So we're very excited to create really the first, what we think is the first good model of CMT2A, which will be used to assess different treatments that we develop. Again, we are using stem cell models derived from CMT2A patients. In this case, we're not making them into Schwann cells, but we're actually making them into neurons. This is a picture from one of my colleagues at University of Wisconsin, where you see these green stem cells here in the middle here. And these red things are just axons that are being projected out from all these cells here within this colony here. So these axons are radiating away from this colony, and these are differentiated motor neurons. So we can use this system, number one, to increase levels of MFN1 and see if we can restore mitochondrial function and improve axonal health. And so that's also a major case here. In this case, we're not turning them into Schwann cells, but actually into motor neurons. So you know the drill by now. We are trying to assemble all the pieces that we need, which includes drug screening assays. This is an effort I've been involved with, and I was just extremely happy. That's why I'm smiling all today. That just in the last week that we finally were able to overcome some technical difficulties and create some assays that have look like they will be suitable for screening at NIH or other partners that we have in our network. This has been a project from my lab, the Baylor laboratory at Cedars-Sinai, and the Ingles lab at NIH. We've developed an excellent rat model that's currently being characterized. So I've shown data from Dr. Shear. And then stem cell lines for CMT2A. We've produced a couple at University of Wisconsin. But this is also part of this major effort through the New York Stem Cell Foundation to create several really good stem cell models for study of CMT2A, which will be differentiated into motor neurons. So ultimately, all of these little pieces of the puzzle are being assembled for all the major subtypes of CMT that we're investigating. And I have to say that as Dr. Shear mentioned, this is not an effort that any one lab or one small group of people could do. I'm going to gloss over names of many people that are involved in this project. We essentially have project teams for CMT1A, for 2A, for 2E, for 1B, and also we're developing approaches for CMT4. So I'm just recapping the slide that Pat showed earlier. I've tried to mention some of the names along the way, but this has been an inspiring effort in terms of assembling with all these different people, both at the clinical end of the spectrum and the basic science end of the spectrum, and everyone who works in between, like Dr. Shear, and to be able to get together and really think about the best ways to attack these types of CMT has been a really inspiring effort. So I wanted to thank you for coming today. I don't know where I am with time. Am I okay? Okay. But that's about the end of what I wanted to present. I'm happy to answer more questions during the career panel, but I don't want to cheat you out of experiencing a presentation for Gina. I was telling Gina I was her warm-up act, so